Today is 21/11 and we are here to revise the biology end of term topics. Topic 1, chemistry in biology. So we have to go through to understand like what do we mean by chemistry in biology. Okay, so that's one. First of all, we have to go through the first KPI that says um, compare van der Waals forces, covalent bonds, and ionic bonds. So three things you have to clarify. There is a very easy thing that you can make a difference between them. So if you see a world that says shared, so like electrons are shared, it means that it is a covalent bond. So the chemical bond that forms when electrons are shared, for example, in water, the electrons are shared between oxygen and the hydrogen of a water molecule. This is called a covalent bond. Now, ionic bond, ionic bond is like when there are forces of attraction. Okay, you don't see a share, you see attraction. Okay, so this is how you will notify it that now this is talking about ionic bond. And these attractions can be easily seen between sodium and chloride, so NaCl, hydrogen and chloride like hydrochloric acid. So these forces of attraction can be seen very easily. If there are attraction, they don't share, but this is a transfer of electrons. So we will say that ionic bond forms when an atom loses and another atom gains. So simply we can say that this is transfer of electron. So we call this one as it is transfer of electrons. There is no sharing of electrons. Okay, so that's how we talk about it. Moving further, we have the last type of the forces, which are the van der Waals forces, and these forces are again attractive forces between the positive and negative regions, pull on the molecules and hold them together. So listen, now here, these are the forces of attraction. These are the forces of attraction, but the difference is over here, you make ions in the ionic bond, but you don't make ions in over here. This is just like to hold the molecules together. Okay, so that's what make the difference between all the three. Just like a quick revision, covalent bond is the sharing of ionic bond is the transfer of electron. Then we move to the last one, which are the van der Waals forces. This is like forces of attraction which hold the molecules together. No sharing and no transfer. Second KPI, so we have to say explain the specificity of the enzymes. So first of all, we need to know that enzymes are protein in nature. They are made up of the protein. They have a very specific site on them, which is known as the active site. So you see over here, these are three enzymes and each enzyme has a specific site. So this is different active site. If you look over here, this is different active site, and if you look over here, this is different active site. So all the enzymes, they have a very specific site. This is known as the active site, and this is the place where reactants bind. So reactant is always called as a substrate. Okay, so substrate bind on the active site, they make an enzyme substrate complex, and in the end the products are released. So this is a product as well as this is a product. Both are called products, and the enzyme is released to go again to do another function. Enzymes, they follow the lock and key model. Enzyme acts as a lock and the substrate acts as a key. So this is how the enzyme substrate works. So these are the only two main KPIs from this topic. I will only revise, boys, the specific KPIs. So let us go to the questions. So now you can participate. First question: which is the correct order of the bond strength from weakest to strongest?
Van der Waals force, covalent bond, ionic bond.
Very good. The answer is D. So van der Waals forces is the weakest, covalent is strong, and ionic bond is the strongest. Where does a reactant bind to an enzyme during a chemical reaction?
Active site.
Active site. Very good. Where does a reactant bind? We got the answer. This is the active site. What is the other name for the reactant? Substrate. Very good. So you have to understand that they can ask as a substrate or as a reactant because these are the same names. Third question, boys. Which of the following or which of the below is not? Remember, boys, whenever you are working on a multiple choice, please follow the directions. Like not. Not means there are 3 correct answers, but one is the wrong answers, and you have to pick the
Light, wrong.
Light, Mister.
very good, light. And there is another factor which affect the enzyme concentration. If it is not light, so another factor that can be here is substrate concentration, and that is a correct factor. What is the minimum or least amount of energy required to produce the products?
Activation energy.
Activation energy.
Good. Activation energy. Do enzymes increase activation energy or decrease activation energy?
Decrease, Mister.
Decrease.
Decrease,
Mister.
Very good. Decrease. Okay. The elements are compounds. The elements are compounds that are made. So made. Who are made?
product.
Products.
Products, Mister.
Okay. Good. Let us discuss all these options. Catalyst. What is a catalyst?
The same as an enzyme.
Mister,
It speed up the chemical
It's
reaction.
This type of enzyme.
Yes, catalysts, they are like speed up the enzyme, they decrease the activation energy, they don't affect the products, but they increase the rates of the reaction. They can be reused in the process again. Enzymes are specific catalysts which are present in living organisms. Good. Product we discussed already. This is the definition. Reactants is the opposite. We have already seen the definition of the reactant over here. So where does a reactant? So reactant is the molecule which bind to the substrate. Okay, and then the active site we also studied in this option. So active site is a place where reactant binds. See how you link one question with another question. So you automatically, if you link a question with a question, you get the answers directly. Okay, let us move now to this question. Use the graph below. Assess, explain the function of the enzymes. We discussed it already.
And they must speed up chemical reaction by lowering the activation energy.
Very good. Now we have the chemical bonds that transfer electrons from one atom to another.
Ionic.
Ionic bond.
Ionic bond.
So we have now second topic, water and solution, and the KPI is differentiate between the two types of the mixtures, which are homogeneous and heterogeneous. Okay, so we have two mixtures. Homo means same. Homo means same, and hetero means different. So it means that when you mix up them and you get a final thing which looks one, it is called as homo. And if you mix some items and when you finish mixing, still you can see different things suspended or different particles which are moving in that, so it means that that is heterogeneous. So first of all, a mixture is a combination of two or more substances. They can either mix and make a uniform composition. They will be making homogeneous. You cannot tell what are the individual molecules added into it. Like if you make a coffee, so you add coffee, you add milk, you add sugar, you add water, but you stir them and you get a coffee. Now, if I ask you, please tell me what I have added in this coffee, you will not be able to 100% tell me because
Homogeneous.
Yes, because it is a homogeneous mixture and you cannot see them individually. Hetero is when you mix many things and you get a final thing in front of you, like salad and biryani, or fog, milk, blood, sand and water. All these examples are heterogeneous mixture. Okay. um so because you can tell very easily, okay, if you have a biryani, you can tell very easily we have rice in it, we have meat in it, we have potato in it, or whatever. You can see them. Hetero is of two types: suspension and collide. Suspension, when the particles settle to the bottom over time, like you mix them, you can see that they are moving, but you leave them for 10 minutes, 15 minutes, and they will be separate and they turn into layers like sand and water. But collide is something the particles cannot be settled. They do not settle, like fog, like milk, like blood. So they are all collide. If you see down, many these pictures tell you examples like tea, sorry, like tea, like juice, medicine, and honey. These are the homogeneous mixtures. And ice in water, soupy noodles, salted candies, salted dry fruits, soil, oil in water. All these are examples of the heterogeneous mixture.
Yes, Mr. There is a machine that can separate blood from plasma.
Yes.
They settle.
Yes. Yes. They settle, but not by themselves. They settle when you apply some force on it. That is called as the centrifugation machine. And that centrifuge, the centrifugal force separate it, but that is by force. Not like generally. If you leave them generally, they may not settle, or even if they settle, they take a long time. Anyhow, boys, this is a table, and this table tells you the difference between a solution, suspension, and collide. Remember, whenever we say solution, it means homogeneous. It automatically called as homogeneous. And if we call suspension and collide, these two we mean as heterogeneous. You got it? Like solution is a uniform mixture, so it is homogeneous and particles do not settle over time because the particles cannot be seen even, so they do not settle over time. Suspension is when they are distinct, heterogeneous, settle over time. Components are distinct. In the collide, they are heterogeneous and the particles do not settle over time. What is common between them? They are all mixtures. This is common between them, and they are all made up of particles. Again, there is a different way. Sorry, there is a different way of examples. So solution, it says that you mix them together like you are adding salt into water. So this will make a solution. Collide is like jelly. You can see over here they made a jelly. There is a fog. Okay, it's written very small. So fog is a collide. A property of the collide is scattering of the light. Now the last one is suspension. So suspension is again a heterogeneous mixture. It separates over time. So like sand and water, oil and vinegar, and sand and water. So these are other examples of the suspension. So please make sure that you read every single thing in this draft, which is even written very narrow. Questions, boys. When you mix oil and vinegar, what does it make? We just did from here.
Suspension.
Very good. Is it a homogeneous or heterogeneous?
Heterogeneous.
Heterogeneous. Very good. Boys, I give you like 5 minutes to read this table word by word. Okay, so we have now the third topic. I hope you have gone through it. So we will be very fast to go. The building blocks of life. the building blocks are like the molecules which make our life, and there are four major molecules. What we need to do is that we need to discuss the name first of all. So you can make a formula CLPN, NPLC, LCNP, whatever, so that you can remember that carbohydrates, lipids, proteins, and nucleic acids. So these are the four major groups of the macromolecules. Then, before going into the function, we must learn their monomer. So you should not always learn the function first. Learn the monomer first. So carbohydrates are made up of monosaccharides or sugar. So if they ask the monomer of the carbohydrate, so you should say that it is sugar or it is a monosaccharide. They only have CH2O. So this is the unit. CH2O is the unit. So you say that they are only made up of carbon, hydrogen, and oxygen. And the ratio which makes CH2O is always 1 ratio 2 ratio 1. You can easily see in the formula, carbon is 1, hydrogen is 2, oxygen is 1. Now we will go back to the functions. Their main function is the energy source. Remember, they are the quick source of energy. Quick, yani the quickest source of energy in your body is always carbohydrates. They are the structural supports in the plants because they store cellulose and they store starch. So they give the structural support. They are the structural supports in hard shells of animals like the shrimps, lobsters, and insects. And the example is chitin. Okay, so I will write them here on the top. So for example, if you say they are the structural support in the plant, example is cellulose. If we say that they make the shell of the animals and insects, so the example is chitin. So these are like stored forms of the carbohydrates. Examples are glucose, sucrose, lactose, glycogen. Even now I can take it there. The cellulose is the example. I can take this one also there. The chitin is the example. Okay, so these are the examples of the carbohydrates. Lipids are made up of fatty acids and glycerol. They are either the phospholipids or triglyceride. And if you remember, the phospholipids, you study them like this. So this is the phosphate group, and you have the two tails, which are like this and like this. So this is a phospholipid, the phosphate group, and the fatty acid tails at the bottom. They are made up of glycerol and fatty acids. Their function is to energy storage. Listen, boys, there is a big difference. This is a quick energy source, and this is energy storage. Okay, no doubt, lipids are also source of energy, but the primary function is the energy storage. They are hydrophobic. That's why they prevent the water loss, or the water doesn't come towards them, and their property is hydrophobic. They provide a biological barrier in many molecules. Examples: fats, oils, waxes, steroids, cholesterol and hormones, phospholipids, triglycerides. So these are cholesterol. All are examples of the lipids. Moving to next, proteins. Monomers are amino acids, and we have, you know that we have studied the formula for amino acid: carbon in the center, and on one side you have COOH, and another side, what happened? Sorry. Yeah, and on another side you have studied NH2. This is how the formula looks like. Give me 1 minute, I will adjust it. Yeah, and then you have studied that there is R on one side, and there is R on one side, and there is H on the other side. So you see that this is the formula of the amino acids. All amino acids, they are made up of a specific unit, which is a repeating unit. Like this is same in all amino acids, and R group is the only different part of the amino acids. Their function is structural support, like nail, skin, hair. You know, these are all made up of the proteins. That's why we say structural support. They are transport substances. They help in the cellular communication. You know that the transport substances, so we study the type of the proteins that is transport protein. So proteins were three
Yes,
different types. So transport protein, they help in the transfer of molecules. Yes, and cellular
receptor
protein.
protein.
Yes, very good. So these are receptor proteins which help in the cellular communication. They speed up the chemical reactions. So they are what? Who speed up the chemical reaction?
Enzymes.
enzymes.
Very good. So enzymes. So we write over here, enzymes speed up the chemical reaction. Boys, this is how you have to actually learn it to get the best marks. See, all these hidden words are not written, but we have studied them. So like transport protein is not written over here, but we studied it already. So that's what you have to focus and understand. Hemoglobin and enzymes are the examples of the protein. There are many other protein examples. We don't need to go into detail right now. Nucleic acids, these are made up of nucleotides, and each nucleotide is made up of three components. And the three components, what we studied, the three components, sugar. I think so there will be a question down, but let's write over here sugar, phosphate, 3-phosphate, and a nitrogen base. So this is how the nucleotide is made up of. And then they are very important to store and transmit the genetic information. They also help to store the energy. Store the energy is ATP, and transport the genetic material is DNA and RNA. This is again another table, boys, for all the same things what we discussed up. Please make sure that you understand it. For example, carbohydrates include monosaccharide if it is only one monomer, disaccharide if it is made up of the two monomers, and polysaccharide if it is made up of many monomers, or it's a long chain of monomers. Then lipids made triglyceride, fatty acids, glycerol. Proteins are made up of amino acids. Okay, nucleic acids are made up of DNA and RNA. Their basic unit is nucleotide. Their basic unit is amino acid. Their basic unit is fatty acid and glycerol. Their basic unit is, all basic units are written at the bottom, and their basic unit over here, it is the nucleotide. Okay, now ATP is also one of the nucleotides, which is a very high energy nucleotide. Next, boys, discuss the structure and functional characteristics of the carbohydrates. We have discussed them already. I will quickly go over it. Carbohydrates made up of carbon, hydrogen, and oxygen in 1-to-1 ratio. And then we have their unit can be written as CH2O. Their monomer is monosaccharide. They can make disaccharide if there are two. They can make polysaccharides if there are more than two. They are sugars, and they are the energy sources which provide also structural support and all this. We studied all in the table. This is for the monosaccharide, this is for the disaccharide, and this is for the polysaccharide. We had discussed about the DNA, that is the nucleic acids. So they store and transmit the genetic information, DNA and RNA. DNA stand for deoxyribonucleic acid, RNA stand for ribonucleic acid. They store and transmit the genetic information. They store the energy in the form of ATP, which is called as adenosine triphosphate, or a high energy nucleotide. They are made up of three parts. The parts are phosphate group, sugar, and nitrogenous base. These are the structures of the nucleic acids and their components. Okay, so we have to We have to see over here all. You can see them later. Question time, boys. I will give you an option to ask another question other than this. Which of the below is not a biological molecule?
Sodium chloride. Sodium chloride.
Sodium chloride.
What should be here instead of sodium chloride? What is missing? Which
Nucleic acid.
Very good. So this is how you recall your knowledge also, that there should be nucleic acid. Good. Nucleotides are made up of 3 parts: phosphate group, nitrogen base, and
Sugar.
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 the monomer of the carbohydrate is
دي مستر. I don't know how to spell it. Monosaccharide.
what else can be written instead of the monosaccharide?
A simple sugar or sugar.
Sugar.
Very
Sugar.
good. Simple sugar or sugar. Very good. So let's write it as simple sugar or even sugar. Okay, good. ATP is what?
Is a high energy.
high acid.
Beef.
Very good. Muscles, skin, and hair are made up of
Proteins.
Protein.
Protein.
Which of the following is the main function of the simple sugar?
Direct energy source.
Yes, they are direct energy source. Okay, let us check quickly other functions. Maybe let's say in the exam they change this part. Okay, they change this part. And if they say that which of the following is the main function of protein?
Structural
Structural
support.
support.
Genetic information.
Very good. Bravo, boys. Amazing. So structural support is for the protein. If in case they change on the top. For which of the following is the main function of nucleic acids?
Storage of genetic information.
Yes, this is the function of the nucleic acid.
And for the lipids
okay?
is energy storage for a long time.
Very good. Amazing, boys. See, now you understand that how one question can be converted into many versions.
Yes.
Like, see, now we have one question, but this can be asked for 4 different types of the questions. Okay, good. Now, carbohydrates are the polymers that consist of the repeating dash monomers and can be dashed to release large amounts of energy.
Sugar broken down.
Good. Sugar and broken down. New topic 2.1 is cell discovery and the theory. Most part of the cell discovery we don't need to understand it, but definitely for the cell theory, yes. So what does the cell theory says? That it has 3 principles. We need to identify like what are the 3 principles of it. So basically whenever it comes as principles, so there are 3. Number 1, all living cells are made up of 1 or more cells. So like we can say that there are some organisms which are unicellular. They are made up of 1 cell, like bacteria, okay, like prokaryotes, and there are like us who are multicellular, so made up of many cells. Second point, it says that cells are the basic unit of life. Cells are the basic unit of structure and organization of all living organisms. Cells arise from previously existing cells. It means that new cells come from old cells. It happens, and also it copy the genetic material, give it to the new cells. So these are the 3 principles of cell theory. Next part is the identify the parts of the light microscope. We don't have the electron microscope. We have here only light microscope. So it has two things, boys. Look, identify. First of all, you need to identify, and then you need to tell the function. So here is a big circle. This one, it is the coarse adjustment, and it is for mainly focusing the image and also move the stage up and down. The small knob is the fine adjustment, and this is to sharpen the image, make it more bright, more clear. Then we have a small hole over here under the stage. This area is called as the diaphragm, and it allows how much light will enter into the slide, and it goes through the objective lens into your eyes. This area is for the support again, and it supports the whole microscope. This is called as the base. I will go back to top to go the second side. So this area over here, this black one is not listed. Anyone knows what is the name for this area?
Mr. Is it tube?
Yes, it is tube, and we call it as body tube. Okay, so it's not listed, but let's write it for more understanding because it is a part of it. So I will try to list it here and then try to link it here. Yeah, so this area is called the body tube, which basically connect the nose piece with the eyepiece. This is the nose piece over here, the circle, this circle, and it is basically to hold the objective lenses on it. And objective lenses can be high power, low power, medium power, okay, and these are for the magnification. These are the small ones are the objective lenses, and you know their powers range from 4x, 10x, 40x, and 100x. 4x scanning and low, 10x medium power, 40x high power, and there is one more 100x, which is called as oil immersion power. Okay, this is oil immersion lens. Okay, so low, medium, high, and oil immersion. This is the stage, and this is for placing the slide on it, and the slide can be held on it by the help of these clips. So these clips are called as the stage clips, which hold the slide. Then we have here bulb source or light source or lamp. It can be any. It projects the light which passes through the specimen, goes through the diaphragm, into the slide, through objective lens, through body tube, into your eye. So that's what you can see the image of the particle. This is the whole microscope labeling and everything, boys. Now let us go to the questions. What part of the microscope sharpens the image?
Fine adjustment knob.
Fine
adjustment.
Fine adjustment.
Good. If I replace this sharpens with another word called magnification.
Course.
Course
Course
adjustment
adjustment knob.
Boys, think again. Which of the following
Objective lens, Mr. Objective lens.
all except the fine adjustment? If you see back, look, we go back. What is written over here for the nose piece? Magnification, right?
Yes.
There is no magnification written with the coarse adjustment. Coarse adjustment is for focus, not for magnification. The magnification is only for the objective lenses and eyepiece.
Yes.
You got it? So if it is magnification, so then you have to say eyepiece magnifies it by 10 times, and objective lens magnifies it, yani 4x, whatever.
Yes.
You understand?
More than one time.
And yes, and if it is a focusing, then it is the coarse adjustment. Okay. Moving next. When focusing a specimen, you should always start with which power?
Low power.
With the low power.
Low power.
Very good. And what is the power for the low? We don't need to actually know it, but it is 4x, 10x, 40x.
40x.
What is the
Cell.
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 The basic unit of the living thing.
Well, very good. Define the three principles of the cell theory and explain them how they were created. No need to explain them how they were created. This part is not needed. Only this part is needed. Yes, anyone, please.
Basic unit of life.
Basic unit
Sir,
of
the basic unit of
living
life
thing.
itself.
Our living thing have cells.
All organisms
Yes,
are
and
consist
the
of
third one.
They are.
All
Cells
new.
arise only from previously existed cells.
Very good. Very good. Okay. Which of the following is not a statement of the theory?
All cells contain nuclei.
Nuclei.
Yes.
According to cell theory, new cells come from.
Dividing pre-existing
Dividing
cells.
pre-existing. Yes.
Very good. Dividing pre-existing cells. No need for this right now because we have done a detailed discussion over here so as to label above picture. And then even for we had a detailed discussion like what are their functions. 3 over here. So the first one is holds and supports the slide.
Stage.
Very good.
Number
Okay.
one.
Let me ask you one more. Here, controls the amount of the light that passes through the.
Diaphragm.
Diaphragm.
Very good. Very good. RAGM. Okay, now the last one from here is projects the light upwards through the diaphragm. Specimen and
Light source.
Light source.
yes, it can be lamp or it can be light source. Okay, good. Let us move down now, boys. Which of the following is an objective lens?
B. Mister.
B.
B is the objective lens. A is the
Eyepiece.
Eyepiece.
yes.
Let's.
A is eyepiece. C
Stage eclipse. Or
Stage clips. Sir.
it's not
Stage
clear.
clips. And D light
Here,
source.
so we can say stage as well as we can say stage clips because it's not very clear. And D
Light source.
Light source.
source.
yes, light source. Very good. Okay, let us go now down to lamp LA. Okay, let us go now down to the next picture. Use the figure below to answer the questions, and we have here functions of the parts, the source of light that is used to view the specimen. Name the
Light source.
yes,
Source.
Source.
The letter is D.
lamp, and what is the letter?
D.
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 Sorry.
Mister.
Yes, D. Thank you. We will say here D. Used to move the stage up and down.
The fine adjustment.
The fine
Mister.
adjustment.
C.
No, it's not fine adjustment who move it up and down.
Source.
It
Course
should,
adjustment.
it should be
Course
course.
adjustment.
Yes.
C,
it should-
Mister.
Yes, it should be.
Mister, but yesterday when I studied this worksheet, it was the fine adjustment.
Let me check 1 minute. I can check over the second page. 1 minute. Give me a minute.
But to move the stage up and down, it's a course. Mister.
Yeah, it should be course. Maybe there is a
Course,
mistake.
sir.
Yeah, I can understand. It is written there, fine adjustment, but the one who moves the stage up and down is the course. You please correct it there, okay? It's fine. It's a minor mistake. And the labeled part is C.
C.
But boys, another thing which we have to focus over here. Look. For example, over here, if you see that it is touching which ring, the bigger ring or the smaller ring?
Smaller.
No,
Smaller.
sir.
It
The
is
smaller
touching.
is the fine and the bigger is the
Smaller is the fine and the bigger is the coarse. It means that this question must have like some issues. It's okay. So try to look into its coarse adjustment and fine adjustment is always like a confusion to get into it. Anyhow, the next topic was the plasma membrane. This is really very important and easy and quick topic. Let us see over here. You have to use the diagrams. So maybe you will be given the Walaikum Assalam. You will be given diagram and you have to label the parts and then you have to write the functions of these. So what we can see over here, phospholipid bilayer. So you always know that from here, from the top to bottom, when you have two phospholipid molecules, you as the phospholipid bilayer. So you see over here, this is a phospholipid, and if there is one molecule, phospholipid. If there are two layers, these are called as the phospholipid bilayer. If you look into it, the head is called as the phosphate head, which is polar. The tail is called as the fatty acid tail, which is a non-polar tail. Even labeled over here, like these are the tails, this area, these are the tails, and these are the non-polar tails, and these areas are the heads, which are the polar heads. In addition to that, you have here these one, this one, this one. These are called as the cholesterol, which increase the fluidity and strength of the cell membrane. On the above surface of the membrane, you have here these extensions. These are the carbohydrates, which are for the signals and cell-to-cell recognition. Now we have very important proteins over here. Very important proteins over here, which are the transport protein. You can easily see these proteins which have a channel between them, like this open area between them. Depends on the movement. Movement is inside or outside. This is a different story, but they are called as the transport proteins. This is the inside of the cell. This is the outside of the cell. So when you have to focus on it, when you are talking about the cellular transport, you have to focus on it. This is another protein which is embedded. This is inside the cell membrane. A little of it is open towards the outside. Next, moving to this diagram, it's again the same thing. It's again the same thing, but just a minor difference of orientation. So you have here the peripheral proteins. It means that only on the outside, and you have here integral proteins, which means they are present from one side and opens to the other side. So they are across the membrane. You have phospholipid, you have glycoprotein and everything. You have again the phospholipid. We discussed it already. And then now we have here the relate the phospholipid properties and water lipid interactions, like the hydrophobic and hydrophilic interactions to the cell membrane. So we know that the polar head is hydrophilic, it's a water loving, and the two non-polar tails are hydrophobic, water fearing, and always the polar heads face outside and the fatty tails face interior of the cell membrane. Again, boys, many different kind of orientations. You have to try to just go over these. Here it also say you that they love water because they're hydrophilic. Tails hate water. They are hydrophobic. And then it also says that there is a fluid inside the cell membrane that makes it a fluid. Yani it designs a fluid mosaic model. And fluid mosaic model, we will say down here, it says something, but let's first discuss this one. Identify the functions of the proteins, carbohydrates, and cholesterol in the plasma membrane. So first of all, cholesterol. Cholesterol keeps moving the molecules. So it means that it maintains the fluidity. It keeps the fatty acid and chains from sticking together and allow the fluidity of the plasma membrane. Carbohydrates for chemical signals and cell recognition. So this is the property of the carbohydrates. And then we have proteins. We have three types of proteins. So receptor proteins, first of all, We have receptor proteins which are only on the outer surface and they help to transmit the signals. We have proteins on the interior which help in the support and the shape of the cell. And the most important of all, we have the most important of all, we have the transport proteins which span through the cell membrane and create a channel for the substances so that they can enter and leave the cell. And this is very, very important for selective permeability. What is selective permeability? Yes, boys.
It allows some to
It
enter the
allows
cell
some
and
to
not
enter the cell membrane.
yes,
Yes,
not all
Yes.
part molecules.
To enter
some.
the cell.
Okay, very good. So it allows some molecules, ions, or particles to enter or leave the cell, but not the others. Not only it allows to enter, but also it allows to enter or leave, but not others. So that's why we call it as selectively permeable. Describe using visuals what is the fluid mosaic model. So cell membrane is described as a fluid mosaic model because its components can move and they are not fixed in position. Mosaic is made up of many parts, and we studied already what are the parts of the mosaic. It's clearly written over here in this one. In this one, they are here. They are made up of proteins, carbohydrates, and cholesterol. So these are the particles of mosaic. It means that proteins, carbohydrates, and these can easily move or float in the cell membrane. Select all the statements that are true or correct about hydrophilic.
All of them.
Mr. All of them are right.
All of the above are right. Very good. So it is a polar group. It is attracted to the water. So all will be correct. There should be an option, basically all of the above. Good. A phospholipid molecule has a head and two tails. The tails are found where?
In the
Anterior.
Interior of membrane.
Who is found at the surface of the membrane?
The heads, Mr.
Head.
Yes.
The polar
Very.
heads.
Where is the environment hydrophilic? Water loving, outside or inside?
Water fearing
Outside.
inside.
Outside.
Water loving
yes,
outside.
yes, outside.
Water fearing inside.
Okay. Let's see over here. Basically, now listen carefully this concept. Basically, the water loving environment is present on both sides of the cell membrane. It is on the outside of the cell as well as inside of the cell. Outside the cell and inside the cell. Because you see the phosphate heads on outside and you see the phosphate heads on inside. So that's why the hydrophilic environment is present on both sides of the cells, but where it is hydrophobic, only in the interior of the cell. Did you understand?
Yes,
Is it clear, boys? Look,
Yes,
I can explain you from here. This diagram. Look, this is a cell. This is a cell. From here, like this. This is a cell. Okay. So this is a cell membrane of that cell. Cell membrane is a high phospholipid bilayer which extend like this. So here is water on the outside of the cell. Here is water on the inside of the cell. So where is the hydrophilic environment?
Inside.
Between it.
inside.
Outside. Hydrophilic outside.
Yes. So both sides of the cell membrane, is hydrophilic.
Yes.
Or consist of polar heads.
Yes, very good. Okay, let us move to the next question now, down where we were here. So At the surface of the membrane is hydrophilic, where the environment is hydrophilic on both sides of the cell membrane. Span through the membrane is the hydrophobic, like the interior. Okay. Use the diagram to label it. We have done the labeling on the top a lot. I'm not going to label it here. The fluid mosaic model refers to
D.
okay. Which of the following is not a function of proteins? Which of the following is not a function of the proteins? So
help
produce lipids. Produce lipids.
okay. The
Mr.
carbohydrates.
answer at number 4, Mr. You didn't answer 4, I
didn't
think.
answer.
Sorry. The fluid mosaic model refers to
Fluidity of phospholipids, cholesterol,
Availability of
carbohydrates, and protein.
phospholipid, cholesterol,
Yes.
carbohydrates, and protein.
Very good. The carbohydrates on the plasma membrane are involved in dash and dash.
Add heat between cells.
Mr. cell to cell recognition and
Mr. Cell
identification
Cell
of
to cell
the
recognition.
chemical signals.
Yes.
And
So these. Okay, good. Explain why the plasma membrane is so important. We have discussed it like it's a barrier. It maintains homeostasis. It is selectively permeable. The structure of the shows below. What?
Cell membrane.
Cell
membrane.
membrane, Mr.
Very good. What is A?
A protein, transport protein.
Mr. A, transport protein.
B?
B, phospholipid
bilayer.
bilayer.
Yes. Okay. So B is phospholipid bilayer. Done. A. We all. What is the role of A?
Mr. Not storage. Channel for transporting certain material.
Yes. Yes. It's a transport protein. Good. Now you have here a table to match, like the column where you can match them easily. So let us go quickly from here. Lack nucleus and membrane organelles. Who?
Prokaryote.
Prokaryote. So that is D. Okay. Helps to maintain the homeostasis.
Cell membrane.
A
Or
plasma
plasma
membrane.
membrane.
No.
Yes, plasma membrane. So that
Cell
is
membrane.
composed of phosphate group, glycerol, fatty acid.
Phospholipid.
Phospholipid.
Phospholipid, Mr.
Proteins that help to anchor cells in internal
Interior protein.
Interior protein.
interior proteins. Very good. So this is B. Okay. Refers to the plasma membrane where substances can move around within the plasma membrane.
Fluid.
Fluid.
Fluid mosaic
Fluid.
model.
C. Very good. Give strength and fluidity.
Cholesterol.
Cholesterol.
E. Okay. So don't worry. Look, there are two extra, but we don't have to answer them. You got it?
Mhm.
Okay. So we have the next article, next topic, which is 2.3. It's again very important. Here we have the organelles and we have their function. And in the second, this one, we have using the diagram. So this is the main difference between them. So you have to use the diagrams basically to write their function. So let's quickly go over here. Ribosomes are very important to produce proteins. Nuclear membrane is the outer membrane that surrounds the nucleus. Cytoplasm is a jelly-like material in the cell, which is very important for all the chemical reactions to take place here. Rough plasmic reticulum mean they have ribosomes and they make proteins and lipids also. Smooth, no ribosomes, and they help in producing lipids and carbohydrates. Also, they help to break down the harmful substances. Cell wall is the outermost protective boundary in the plants and the bacteria, but not in the animals. So remember, this is only in the plants. It gives support. Vacuoles, large vacuoles in plants and small vacuoles in animals. They are mainly for the storage for food, water, and enzymes. Nucleus is control center of the cell. It's the brain of the cell, okay? And it contains the DNA. It controls all the activities. Cell membrane, we study the whole topic about the cell membrane. that it is a protective covering of the cell, and it allows some molecules, like it controls what enters or what leaves the cell. Nucleolus is present inside the nucleus. It's a small part that produce ribosomes. Golgi apparatus, it modifies, packs, and salt the proteins where they will be given, like whether the outside or inside, and they transport it by the help of the vesicles. What are the vesicles? Vesicles are tiny sacs that contain and transport proteins and other substances outside the cell. Mitochondria is the powerhouse of the cell. Lysosomes, because they produce energy, by the way. Lysosomes are the vesicles, but these vesicles contain very specific enzymes to digest and break down the waste materials and the dead organelles. Chloroplast, only present in plants, help in photosynthesis, and this is very important to convert the light into chemical energy. Cytoskeleton is made up of protein, and they are made up of long, thin proteins of fibers, and they make a framework of the cell to give it a shape, to anchor the organelles, and to help in the movement. Centrioles are only present in animals, and they are very important for cell division. There are 2 centrioles present in each cell made up of microtubules. Cilia, small hair-like structures. These are short hair-like structures, and they are present on the outer surface of the cell or inner surface of the cell, and they help in the movement.
It's
Flagella.
like the flagella. This is like the flagella, right? But
yes.
short.
Yes, true, but short. Yes. So flagella is a long, and they are present on the surface of the cell. Mainly the cilia are on the inner surface of the cell, and flagella are on the outer surface of the cell, but both help in the movement. Okay, so now here are the diagrams. Functions, we will not go into details. You can read the functions if you want, but I will just check the diagrams quickly. I will zoom, and let's see. Cell wall, this area is the cell wall. Centrioles, you see them like this. Chloroplast, you see them like this green, and the coins inside, the piles. Cilia or flagella, both looks like this. And cytoskeleton, it's not clear, don't worry about it, but it's the internal framework of the cell. ER are like this, they can be rough, like these are rough, and these are the smooth, okay, outer side. The close to the nucleus are rough. Flagella, I told you, cilia and flagella both are same. So you see the arrangement, cilia and flagella both are same. Golgi bodies, we studied that they look like Wi-Fi signals. You see them like this, and then you have one more like this. These are like the Wi-Fi signals. Lysosomes are the vesicles, empty vesicles, but they are filled with the enzymes which break down and digest the dead organelles. Mitochondria, they have projections like this inside. The difference between the mitochondria and chloroplast is mainly this one. Nucleus, the brain of the cell, and this area inside, this one is nucleolus.
Yes, Mr.
Yes, produces ribosomes. Yes, we studied this. This is a cell membrane. Ribosomes look like this. They are made up of the 2 units, one bigger unit, one small unit. Vacuole is like this. It's a large central vacuole in case of the plants. Okay, so these are how their diagrams look like. Quickly going into the cells wise, the red arrow shows what the organelles for me. The red arrow is what?
Nucleus.
Nucleus.
The brain of the cell that
Mr.
control all
The brain that controls.
The brain, Mr. That control all the process.
Powerhouse of the cell.
Mitochondria, Mr.
Mitochondria.
Yeah.
Very good. Who is the protective boundary of the cell?
Cell membrane.
cell wall.
Cell membrane or
Cell membrane.
Yes, cell membrane in animals and all other cells, but cell wall in the plants. But because this is an animal cell, so we will write a cell membrane. The storage house of the cell.
Vacuole, Mr.
Vacuole.
Vacuole.
Yes, vacuole. And the upper one was nucleus. Okay, let me just write that. So this one was nucleus. Okay, moving down. Okay, moving down. What is this organelle? Name
Chloroplast.
it. Chloroplast.
Chloroplast, mister.
How you check
B.
the function?
Mister, it's the principle of the process photosynthesis.
It is found in the
It is
only
found in
plant cell
the
and helps
plant
in the
cells and help in
photosynthesis.
photosynthesis.
See.
This is C. Okay,
C.
good.
Yes.
What is this organelle?
Ribosomes.
Ribosomes,
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 protein
Protein synthesis.
ribosomes and
Very
protein synthesis.
Very good. Cell structure that participate in the digestion of dead.
Lysosomes.
Lysosomes.
Isomos.
Lysosomes.
Lysosomes.
Very good. Identify the number in the organelle that modifies, packs the proteins.
1 1.
One.
One.
What's the
Golgi.
name?
Or
What is
Golgi.
the name? Golgi. Very good.
Yes.
Because
Yes.
it's like Wi-Fi signals.
Yes. Very good. Golgo, not Golgo. Golgi. Okay. Next, organelles which are present in all types of the cells and they are very
Ribosomes.
Ribosomes.
Ribosomes, mister.
excellent. Okay. Next part, boys. This topic is very, very important because it has one period extra as compared to the others. So focus more over here. If
Your voice is lagging.
Mister,
No,
it's
Mr.
not.
His voice is clear.
no problem.
No, it's not.
Abdullah Jafar, it's always same, always in your own periods, in your own class periods also it's the same. You always say me the same and I reply always, Abdullah, you need to check your voice.
Sorry, Mr. Sorry.
Okay, yes boys. So we will make it as quick as possible. Cellular transport is like how the molecules move in. We just need to focus on two main types. The two main types are the passive transport and active transport. So first of all, the passive transport is of three types: diffusion, facilitated diffusion, and osmosis. Remember boys, simple diffusion or diffusion is when any particle move from high concentration to low concentration. So passive does not require energy and they move the molecules from high concentration to low concentration. All passives, whether it is diffusion, whether it is facilitated diffusion or osmosis. Now, so first of all, diffusion from high to low, okay, they don't need energy and the examples can be any. Facilitate mean to take the help from someone. Someone are who? Either they are the channel proteins or they are the
carrier proteins.
yes. Again, boys, no need of energy. Just the need is channel protein or carrier protein. And remember again, the movement is also from high concentration to low concentration. Okay. Osmosis is only and only for water. Okay. And that is also from high concentration to low concentration. Okay. Now in these pictures, you need to identify them. How you say this is osmosis, because it's written over here. This is what?
Water molecules.
Water. Very good. And also the shape of the molecule, try to get it. It is H2O. So you have to identify, maybe this water is not written, maybe, but the shape shows you this is water. And also they need aquaporins like the channels, and also they can move through slowly, or they cannot, like this arrow shows they cannot, but from here aquaporins, yes. Over here it's a simple, but they are using the help of the protein channels. So what is this? Channel mediated. They are taking the carrier molecule supported, so it's facilitated diffusion again, and it is simple diffusion. So try to identify the pictures. Same here, boys. Try to identify the pictures. Now you don't see the water written here in this picture. Look into this picture. You don't see water written, but you see the water molecule shape.
Yes, it's oxygen and turkey.
Yes, or you see aquaporin. So if you see aquaporin or you see this, or you see water written. So anything you see, it means that this is osmosis. Okay, fine. We have now to distinguish between hypotonic, hypertonic, and isotonic solution. What do we mean by hypo?
swollen,
Hi.
mister. It gets bigger.
Yes, Mr. It gets bigger.
Yes.
Yes.
Okay, let's focus on this picture and also keep the definitions on the top like this. Okay, so hypo mean, first of all, tell me the meaning of hypo.
Less, Mr. Less.
Yes, low or less.
High. Low to high.
Tonic, what is tonic?
Hypertonic, Mr.
No,
High
tonic,
to low.
tonic, only tonic. Tonic means solute. Okay, so it is a
Understood.
solution in which there is less solute. So the cell will, what will happen to the cell?
Will increase in shape or size.
Yes, it will swell.
Increase, mister.
Yes, very good. And also, boys, look, there is a quick help in the picture. Can anyone identify? There is something special on the picture which help you to tell if the water is going in more or water
Mr.
is going out.
arrows,
The
Mr.
arrows,
Because of the
Mr.
The arrows,
molecules,
Mr.
not
The arrows.
high,
The arrows,
Mr.
the arrows.
Yes, bravo. The arrows. So count the arrows here. Look, 1, 2, 3, 4. 4 arrows are going in, but only 1 and 2
Two is
arrows are
going
going
out.
out. It means that more water is going into the cell. Cell will swell, and it is a hypo.
Then
In the second picture, it is hypertonic because also, boys, one more thing. What does these dots show? These dots.
The water is going inside.
molecules.
Molecules, very good. And these are called as solute particles. These are called what? Solute particles. So it means that the solute is more on the outside. If the solute is more on the outside, definitely the water will leave the cell. And also you see now the out arrows are more. 1, 2, 3, 4. So water more comes out because the solute are more on the outside and the cell will shrink, and this is called as the hypertonic solution. How you decide this is isotonic?
Mr.
They are the same number
the molecules
of
and the
inside
arrows.
and outside.
Yes, the molecules are almost same on in and out, and also the arrows, 2 arrows going in, 2 arrows going out. And the third thing, you don't see any change in the shape as compared to the first and second picture. As compared to the first and second picture, neither it is swollen in the sea nor it is like shrink. So that's why this solution is isotonic. Okay, that's good. Let us move to next. Again, the same thing, boys. You need to identify now from different picture. The cell is swollen and it burst, means lyse. So this is a hypotonic. The cell is normal. So this is isotonic. The cell is
shrinking,
shriveled, and this is hypotonic. Plant cells become turgid. Yes, okay. The plant cells become turgid. Turgid means swollen. More water is going in, so the plant will swell, but it will not burst because it has a cell wall. So because the plant is swollen, this is called as the animal was swollen, so this is a hypotonic. The plants shrink. In the third picture, the plant cells shrink. So when the cells shrink, this is hypertonic. In the center picture, there is a normal animal cell and there is a normal plant cell. So that's why this is isotonic. Okay. Now the last part of the topic, which is active transports, we discussed it already. It needs energy and it is from low concentration. It is against the concentration gradient. It is against the concentration gradient. It means that from low to high concentration. It can be either with the help of the protein pump, it can be endocytosis, or it can be exocytosis. And endocytosis is like when the large molecules move into the cell. You can take different pictures to identify the molecules are arrows, boys, arrows. Always focus on the arrow if there is any arrow. This is from low to high. So what it is?
اكتب مستر.
اكتب مستر.
active transfer energy.
Yes. This is from high to low.
Diffusion.
This is from high to low, but you
Water
have a
molecules.
Have
Yes.
water molecules, it
Process
means
of
it's osmosis.
Osmosis.
very. You. What is this picture? This picture. Which type of movement? This one.
Transfer.
Facilitated.
Osmosis.
Facilitated diffusion.
Okay.
Because it's
someone-
using the help of protein. مستر.
From the low to high.
Okay, okay. It is moving from low to high. I agreed because the arrow is going down and downside is high. The arrow is coming from here, which is low. So it is moving from low to high. It is using the help of the protein channels. It is the carrier molecules. So it is
Yes.
also. But what about this?
Energy.
Does facilitated diffusion use energy?
No, mister.
No, it is an active transport.
Active
It's
transport.
ATP.
Yes, yes, energy is ATP, and you have to focus that it is active transport, boys. Look, you couldn't identify the energy. I always tell you if there is energy, so make sure that you directly go to the active transport. Yes, also it was from low to high. This picture, boys, the arrows again, arrows are going in. So what is this?
Endocytosis.
Endocytosis.
Arrows are going out.
Exocytosis.
Exocytosis.
Very good. And how you get the arrows are going where? You have to look over here. This is extracellular environment. Extracellular mean outside the cell. Okay? Extracellular mean outside the cell. So how you get it? Where is inside and where is outside? From the labeling. So this tells you outside the cell. Okay. Now from these pictures, boys, quickly, just tell me the solution. Which solution is hypotonic?
B.
A.
Hypotonic. Yeah,
Low to high.
hypotonic.
Hypotonic is A. Hypertonic is
And
B.
hypertonic is B.
Hypertonic is B. Very good. Okay. Diffusion is what?
Solvent moves.
Diffusion. Molecules move from
Mr.
which
move
Molecules
High
from
from
area
of
high
to
to low.
high to low. concentration.
Very good. Active transport.
Requires
all of
energy,
the
Mr.
above, correct.
All of the above, correct.
Yes, it requires energy. It requires a transport protein, and it also moves the molecules against the concentration gradient and against mean from low to high. When a cell is placed in a hypotonic solution, so what will happen to the cell?
Water enters cell toward the area of high solute concentration. Higher frozen.
Very good. What process do cells use to get large substances into the cell?
Endocytosis.
Endocytosis.
Very good.
Endo-
Use the
Go inside the cell.
water is going in.
Hypo.
Hypertonic.
Very good. Hypo.
Hypotonic.
Excellent.
Because it's getting bigger.
Yes.
Pop.
Yes. A type of the endocytosis in which cell ingests solid.
Phagocytosis.
And when it is liquid,
What is phagocytosis?
Yeah.
when yes,
Mr.
pinocytosis. Boys, when look in this picture, the molecules are taken into the cell, inside the cell. It is called as endocytosis. But if the molecules are solid, this will be called as phagocytosis. If the molecules are liquid, this will be called as pinocytosis. Exocytosis is moving the molecules out. Osmosis is only the movement of water. What is shown by this picture, boys? Which type of movement?
Active
Osmo-
transporters
osmosis,
molecule
osmosis.
needs energy to move.
Osmosis, Mr.
Very good. Osmosis. Very
Because
good.
water.
Then osmosis has two solutions spread by membrane.
Yes, so it is osmosis as it has two solutions separated by a semi-permeable membrane. Very good. Which of the following is true for the passive transport?
Need energy
It works
water
along
No.
the
1, 2, 3,
concentration
1, 2,
gradient
3.
2 and
below
3, 2 and 3, Mr.
between
C.
the
Mr. 2 and
carrier
3.
molecule.
Passive transport
C.
doesn't
See,
need energy.
Mister, it does not
Only
need
active
energy,
transport
Mister.
need energy.
Simple
Yes.
and the F1, Mister, and osmosis no need
They
for
need
energy,
extremal
Mister.
acceptance. Yeah.
Very good. Very good. Transport. If I say active
One
transport,
only.
one only because
Yes.
it
It
needs
don't need carrier.
it needs carrier molecule, but not sometime. Sometime you can say yes. So it is 1 and 3. When you say the active transport, that will become 1 and 3. If you say passive transport, it will become 2 and 3. Okay, moving to next. Just say me the
Mister,
name
can
of
you
the
repeat 8?
8.
Yes, Mister.
8,
Because I wasn't
8. 8. This one is I ask you identify the process. So you said it is osmosis. Why? Because first of all, you see it is the movement showing in the water molecules. Okay, you just see the water molecules. Look,
Yes, Mister.
these dots. Count these dots. 1, 2, 3, 4, 5, 6. Okay, count over here
Yes.
also. Count over here.
6 also, Mister.
So it means that the molecules are not moving. So who is moving? The liquid. First, the liquid was here. Now the liquid is until here. This is the final. See? So it means that the liquid moved. And in which process you studied the movement of the liquid?
In osmosis.
Osmosis. So that's why this is osmosis. And also it needs a semi-permeable membrane like the cell membrane. So that's why we say that this is osmosis. Okay?
Okay.
Question, I don't need the explanation a lot over here. Just tell me the process. Identify the process.
Active transport.
Active transport.
Yes, Mister, because there's ATP, Mister.
Yes,
And
excellent.
low to high, Mister.
Yes, excellent. And it is active transport because it is using ATP. Excellent.
And low
Okay.
to high.
Transport. Okay. Transport. Okay. Next question, boys. You see over here, the plant is shrink. Now the plant is swollen. So what was the solution over here if the plant is shrink? Which
hypersonic, Mr. Then it became hypotonic.
Then it became swollen, so it means you gave it hypotonic solution, and the process for the movement of water is what? Osmosis. Okay, so this is how it.
Mister.
Identify the picture, boy. Here, identify the picture.
Ink for the
Simple diffusion.
Simple diffusion. Bravo. Anyone who
Yes,
said simple diffusion, it is excellent because you just see the movement of the particles. This is the first stage and this is the last stage. They are totally.
Mister. This is water.
Yes.
Osmosis water.
Again, okay, listen.
It's water, but it's not the movement of the liquid, or
Yes.
it was the movement of the particle, so that's why it's simple diffusion.
Excellent. You see, I will take you back to this picture. This picture. It
why
was
not
also
facilitated, Mister?
1 minute. I will go down 1 minute. In this picture, there were molecules and there was water as well. But who was moving only?
Particles. Low to high.
In this one, water was moving only. So that's why we said this is osmosis. Now go down, and in this picture, we see the level of the water, all same. And also, there is no semi-permeable membrane shown over here. There is no semi-permeable membrane shown in this picture. That's why it is not osmosis. Now, someone of you ask, why not facilitated diffusion? Because there is no carrier molecule or the channel protein shown in this picture. You got it?
Yes.
Yes,
Yeah.
Master.
So it is simple diffusion because the molecules are moving from one side to another side, and then they reach to one dynamic equilibrium. Okay, let's move to next. Which of the following best describe facilitated diffusion?
It's the movement from high to low connection with help of channel protein. A.
Concentration with help
Very
of A, Master.
Century.
excellent. A. Good. Okay, use the figure to identify the type of the cellular transport across the roots membrane. So now you need
Active transport.
Passive transport.
Low to high.
Yes, sir.
because there
Because
is
low
low
to high.
concentration and there is a high concentration inside the cell already. Okay, so that's why we say it is an active transport. Very good. Diffusion is the movement of particles, especially the gases, and which factors affect the diffusion?
Temperature and pressure.
Pressure.
Yes, temperature and pressure. Okay. With that, the topic is over. We move to 3.1. We are very close to end now, and inshallah it will be like maybe 20-25 more, and then we will be done. 3.1, plant cells and tissues. In this one, the first two KPIs which are very close to each other, and it says that identify the major types of the plant tissues to include meristem, dermal, vascular, and ground tissues. It means that there are 4 types of the tissues we have to identify, and then we have to say their functions, but it can be diagram as well as it can be explanation. So again, there are 4 types of the tissues. These are the 4 types of the tissues. We will go one by one to each. I will try to make it as simple as possible. Meristematic tissue is like actively dividing cells, boys. Remember this thing. They are quickly dividing cells. So where are the cells quickly dividing? In the tips. So if you see this world, meristem tissues at the tip of root and shoot, it is called as the apical meristem. Now, intercalary, it is in the growth on the sides of, you can say, in the nodes, or they produce new cells to result in increase in the stem and leaf length. Okay, lateral meristem, it increase in the root and stem diameter. So now it is clear. Apical meristem, it is actively dividing at the tips. Intercalary meristem, it is mainly at the end in the leaves and in the stem, and the lateral meristem, it is mainly increasing the diameter of the plant. So also one more thing, apical meristem, it is responsible for the primary growth, and the lateral meristem, it is responsible for the secondary growth. Okay, this is another difference. Types of the lateral meristem, we don't need to really go in detail, but anyhow, we have a vascular cambium, which is like for the transport. We have a cork cambium, which is on the outer side of the cell, like the outer protective covering. Yes, it's like the outer protective covering of the plants and the cells. Next tissue, boys. Dermal tissue. Derm for the skin. It means that the dermal tissue is the type of the tissue that make up the outer covering of the plant, and that is called as the epidermis. The dermal tissue mean the epidermis, the layer of the cells that make up the outer covering of the plant. It is called as the epidermis. Okay. Most epidermal cells can secrete a fatty substance which is called as a cuticle, which is on the outer surface of the cell. It protects them from water loss, it protects them from insects, it protects them from other harmful like maybe by eaten by the animals. So there are many functions of it. Stomata. Where are the stomata present? In the lower epidermis or upper epidermis of the leaf?
lower mister.
upper
Very-
epidermis.
lower
They-
lower مس.
lower
Let me clarify you. You both are correct. They are a lot in the lower epidermis, very few in the upper epidermis, very few. So that's why we say that the stomata are present in the lower epidermis and they are supported by two guard cells. They are supported by two guard cells. So these guard, what is the meaning of the guard?
protect
Protect. Yes, very good. So the guard cells protect and regulate the opening and closing of the stomata. So just like the guard, they open the gate of your house, you know. So same way these guard cells, they open and close the stomata to make sure there is not a lot of loss of water. Trichomes are special epidermal cells which are present on the leaf and they are hair-like structures. They give leaves a fuzzy appearance and can help to protect them from insects. Like they're like spikes. You see over here, they are like spikes. So on the leaf, these are called as the trichomes, even the bigger ones.
مستر but
Some
they're so tiny.
Yeah, they are very tiny. Yes, we cannot see them normally. We ignore them. True. But if you touch a leaf and you feel it like it's spiky, it means that these are the trichomes. Root hairs. We did it. Okay. The function of the root hair is to increase the surface area of the root, so make them able to absorb more and more water from the soil. Vascular tissue, boys, very important, which help for the transport of water and food. We studied already. Xylem helps in the transport of what?
Water, Mr. Water and filament.
Water carrying vascular tissue.
Very good. And it is made up of what? Vessel elements and
tissues.
Element and trashes.
tracheids. Okay. Now, what are the vessel elements? They are basically like structures which are joined end to end to make a long opening, and the tracheids are like cylindrical cells. So both of these make xylem, and xylem is important for the transport of water. You can see in this picture, these are tracheids, and here in this picture, these are the vessel elements. So one vessel element from here to here, and then the second vessel element from here to here, and so on. And both of them, they join together to make an hollow opening from inside, because the xylem is dead. Xylem is dead. It's not living. So, and it's hollow from inside, so more water molecule pass through it. Phloem is living because it has companion cells, and these companion cells contain nucleus. So that's why it is living. And the function of the phloem is to carry the dissolved sugar and other organic substances throughout the plant. They are made up of sieve tube and the companion cells, and they are like very important because the nucleus gives them energy and they move the molecules with the help of energy. Ground tissues are the last type of the tissues, which are made up of all three types of the cells: parenchyma, collenchyma, and sclerenchyma, and their functions are in the photosynthesis, storage, and support. You know that like parenchyma mainly for photosynthesis, and the collenchyma mainly for storage, and the sclerenchyma mainly for the support because they are dead. This is another type of the flow chart, boys. You can easily go through it, like types of the plant tissues: meristem, dermal, vascular, and ground. And then what are their further types and what are their further types? You can see them. Questions. Which of the following type of the tissues is made up of vessel elements and tracheids?
xylem sir.
Xylem, Mr.
Xylem.
Which type of tissues allow the movement of water and nutrients?
Vascular.
vascular
vascular.
Yes, vascular bundles. Very good. Okay, 1 minute. Sorry. Vascular tissues. Okay. But if you have to go for only nutrients, only nutrients. So what it will be?
Ground.
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 No, not for. Sorry, not for this question. Like I ask you, any type of the tissue, the type of the vascular tissue which help in the transport of nutrients.
دي مستر.
Miss, that is
Okay, look, the movement of water is done by a type of the transport tissue, vascular tissue. What is the name of vascular tissue?
Xylem.
Xylem.
The
Yes,
xylem and the phloem.
xylem. Okay, and who transport the nutrients?
Phloem, Mr.
Phloem. Yes.
Phloem.
Okay. Next question, boys. Which meristematic tissue is responsible for continued growth of plants after being cut or trimmed? Cut or trimmed?
Apical
خليها
meristematic.
No,
C,
boys.
C.
This should not be apical.
Lateral, Mr. Lateral.
دي مستر lateral.
Again, boys,
Intercell.
listen. Umar? Umar,
Yes, Mr.
can you clarify why did not you choose apical meristem?
What, Mr.?
Why you didn't choose apical meristem?
What, Mr.?
Any other boy? Why apical meristem is not answer for this? Boys, what happened? Apical meristem. What hint I gave you for apical meristem?
Mr. The
Mr.
tips.
Stems.
Tips. Tips of the root and
Yes.
shoot. It is for the apical meristem. Lateral was when they increase in diameter.
Yes.
And when you cut and trim the bush, or bush mean, you know, the woody plants, small
Yes.
woody
Yes.
plants. So
Yes,
that's
Mr.
why.
Mr. Sorry, it was disconnected.
Yeah, I checked you. I saw that you are not there. That's why I started to discuss. It's okay. Which of the following does not match?
Ground, Mr. A.
Vascular
Vascular storage.
Vascular tissue storage.
Yeah, vascular tissue for the storage.
Sorry.
Ground
Yes, Mr.
tissue for photosynthesis, it is true. Dermal tissue for plant covering, it is true. And meristem for producing new cells, it is true. Vascular
Ground for
tissue,
photosynthesis, Mr.
yes, I will show you why. Look, ground tissue is made up of parenchyma, and parenchyma is responsible for what? For photosynthesis.
Yes.
Mr.
Collenchyma for storage.
Yes. Okay. And over here, the vascular tissue is responsible for the movement of water and the nutrients. But here it is written for the storage. That's why it is wrong. Okay. 3.2, boys, root, stem, and leaf. We have to just know about the sequence of the tissues through which the water passes. So first of all, you know that there is root here. Then you know that there is epidermis, then cortex, then epidermis. Sorry, epidermis. There is another one, boys, before xylem. Who remembers?
Phloem.
Pericycle.
Pericycle.
Yeah, it was pericycle. So I will label it as number 5. Okay, it's missing over here. So let me just quickly label this as number 5. And then we have xylem, or the vascular tissue. So that is number 6. Okay, so root here to epidermis, epidermis to cortex, cortex to endodermis, then pericycle, then xylem. Maybe you don't have the root here, okay, because the question already says you from root here to the xylem. So you basically need to know all these 4, because the last one definitely it will be xylem, and the first one 100% it's going to be the root here. So basically you need to know these in between. What's in between? The figure you can easily get over here. Look, root here, then epidermis, then cortex, then endodermis, then pericycle, and then xylem inside. And this is again the same sequence that we just discussed. This is the root here, this is epidermis, then cortex, then endodermis, then pericycle, and then you have the xylem. Okay. Now 3.2.6 describe the leaves characteristics and adaptations. So it means that you need to understand the structure of the leaf and you need to understand the function of the leaf. Okay, so first of all, characteristics of the leaves. It has a leaf blade and it can be simple or compound. It can be like organized in different types in the plants. If they are like simply opposite to each other, or they are called as opposite. Sometimes they are alternate. Sometimes they are whorled, and the arrangement of the veins in the leaves is different. And there are two types of the plants, like monocots and dicots. So monocots, they have the parallel veins, while the dicots, they have the branched veins. Anyhow, this is a general information. Our main concern is the modification, this one. So this is like the one which we have to focus more. So how the leaves are modified? The leaves are modified like in the cactus. They have spines. They don't have the leaves. This is because they need to store water
and protect.
Yeah. Yes, and protection from who?
From animals.
Animals.
Yes.
So, by animals.
Yes. Also,
So
they
that's
need.
why they have spikes, mister.
Yes.
But big spike, not like the leaf ones, mister. That called the start of the T, mister.
Yes. Yes. True. Let us move to the next part of it. That is explain how the structure of the roots, stem, and leaves are adapted to their functions. So you have studied it already. Like roots, they contain a root cap. And this root cap is for the protection, basically, because the root has to go down into the soil. Then we have epidermis, it is the outer covering. And then we have the cortex, which is like mainly the ground tissue, and it's for the transport and storage. Then we have the endodermis, which is on the interior protection, and it's like arranged in a circle, and it has waterproof strip that is called as the Casparian strip, because that is a barrier between the outer and the inner surface. So it helps in the in the barrier force to protect and to stop the minerals to come out. Then we have the last one, that is the pericycle, which produce the lateral roots. Stems. The main function of the stem is to support a plant and produce flower. Vascular tissues are present in the stem, so that's why it is also important for water, movement of water and dissolved substances. And then also these vascular tissues are arranged in bundles. This picture is for the leaf. So you have to look into the structure. You know that the leaf outer surface on the top, this on the top is called as the upper epidermis. Remember that we just cut the leaf and we are looking into this area. Okay. On the upper epidermis, we have a cuticle. This layer, the white layer, which is the waxy and protect the water loss. The below layer, this whole layer on the down is called as the
lower epidermis.
Lower epidermis, very good. Look, also the lower epidermis has a cuticle, but that is very
thin.
less,
Yes.
just like nothing. We have stomata on the lower epidermis. Why it made the heart? Wait. Okay, so we have stomata at the lower epidermis, and it is supported by the two guard cells. So whenever the stomata are open, the gases are exchanged, the water vapors come out and in, and the guard cells, they protect them when to do this and when not. This circle in the center,
Vascular bundle.
Yes, this is vascular bundle, which is made up of two. What are the names of the two vascular bundles? What are the vascular bundles?
Xylem and
Yeah.
phloem.
And phloem.
Yes. Okay. Then there are airs. Okay. Now we have to look at this layer, boys. This layer. This one. This layer. This is very packed and long cells. They are called as the palisade mesophyll, and this is the place where most of the photosynthesis takes place. Why over here? Because it contains a lot of chloroplast, and this is the area where maximum light is absorbed. So leaves do photosynthesis, but which layer is mostly involved in photosynthesis, boys?
Palisade mesophyll.
Yes, palisade mesophyll. Very good. Now only one left, which
Spongy.
is the spongy, and this is mainly for the gas exchanges.
Yes.
Very less photosynthesis takes place here, but there are another thing that is the air spaces. So you see over here, these are the air spaces. So gas exchange is very common in these air spaces. Okay, so what is the main function of the leaf?
Photosynthesis.
Very
Yes,
good.
Mr.
And
Host.
which part of the leaf is mainly involved in photosynthesis?
Palisade mesophyll.
Yes, palisade mesophyll. Which part is mainly involved in exchange of gases?
Spongy
Sponge,
mesophyll.
Mr.
Sponge.
Very good, very good. And who contains stomata?
Lower epidermis.
Or epidermis. Bravo. Okay. Which plant part is not our part of the root?
Stomata. Because it's in the leaf.
Stomata.
Yes,
Yes,
Mr.
Yes.
it is in the leaf. Very good. One of the
Add
main
stem, Mr. Not only leaf.
Store water.
Carry out
Transport.
Transport
Transfer.
and water between
Transport
roots and
water.
leaf.
Yes.
Very good, very good. Which of the below is not of the root?
Increase the length.
increase the length of
Increase
the
the
stem.
Okay. All these questions, boys. Look here. Which part is not a part of the plant? Maybe next time they ask you which of the below are the parts of the root. So you should know that these three are the parts of the root. Same here. What are the functions of the roots? So these three are the functions of the roots. Okay. Now, sequence the layers that the water passes through. We studied over here the sequence.
Epidermis,
Versus
cortex,
epidermis.
endodermis,
Root hair,
predis,
epidermis, cortex,
bredicycle, and
epidermis,
vascular
cortical, and
cambium
vascular.
produce exam and folium.
Excellent, excellent. Okay, which part of the leaf carries the most photosynthesis?
Placid,
Placid mesophyll.
Okay, now look, I can change the same question 4 times again. Which part of the leaf protects it?
Mr.
sponge,
Cuticle.
Cuticle.
Very good. Which of the following is the outermost layer of the leaf?
Epidermis.
epidermis,
Which of
Mr.
yes, Ahmed.
The screen has stopped on me. I will leave and I will join back.
Okay. Which part of the plant, which part of the leaf carries the most of the gas exchange?
Spongy mesophyll.
Spongy, Mr. Spongy.
Very good. Okay, next question. The labelings. What is A?
Mr. Upper epidermis.
Upper epidermis, Mr.
B?
Palisade mesophyll.
Palisade.
Palisade mesophyll.
Very
Yes.
good.
C vascular bundle.
C?
Vascular.
Vascular bundle. Okay.
D spongy mesophyll.
Mr. Spongy mesophyll.
Very good. And E?
And
And
E
then,
lower
Lower
epidermis.
Oh, good. That's great.
The last one is stomata.
Okay, 1 minute. E is, oh, I better copy this one. E is the lower epidermis.
Mr. The last question
Yes,
is
sir.
stomata.
lower of them.
Which leaf structure allows the water evaporation? This is stomata.
Stomata.
Yes.
Yes.
Yes,
Right.
Mr.
Okay. Second last topic wise, this is the second last topic, and it is very easy topic: plant hormones and responses. So first of all, we need to know that there are the 5 plant hormones that we studied: auxin, gibberellin, ethylene, cytokinin, and abscisic acid. So what we need to know now, one by one, quickly, we will say auxin. We will highlight the main functions which are already done in the this light pink blue color here. So auxin, it stimulates the length of the cells. It means that auxin helps in the tip's growth. Either it is the root tip or shoot tip. It helps in the tip growth, and it is also involved in the apical dominance. Also, it helps in the fruit formation and inhibit the dropping of the root. But remember, the auxin, it promotes the cell growth and inhibit the root growth. This is a very common concept that we need to know. If there is a low concentration of the auxin, it stimulates the cell elongation. But if it is in a very high concentration, it will do the reverse effect. Reverse effect means it will stop the cell elongation if it is in very high concentration. It means that the auxin works in low concentration to stimulate the cell elongation, and it causes apical dominance. But if it is a very high concentration, it will make the plant short. It will not let them grow. Gibberellin, which is like for the cell growth, for stimulate the cell division and affect the seed growth, and it helps in the height. So remember, if any plant is short and you need them to be tall, so you need to give them gibberellin. So gibberellin, like here, high concentration of the gibberellin can let the plants to increase in height. Okay. Ethylene is a fruit ripening hormone. Okay, and it is a gas. It can diffuse through the spaces
Yes.
between cells. It is the only hormone which is present in the gas, and it makes the fruit softer and sweeter.
Sweet. And change its color.
Yes. Cytokinin is for C. C for cell division. It stimulates the production of the proteins needed for mitosis. Mitosis is a type of cell division, so it increases the rate of the growth. There is one more which is called as abscisic acid. It's not written over here, but abscisic acid is the most hormone which inhibits or limits the cell growth or the plant growth. The presence of other hormones, especially auxin, influence the effect of cytokinin. So it means that auxin is working with cytokinin. For example, auxin alone, IAA stands for indole acetic acid. You don't need to learn it, but remember that if you see IAA, it means auxin. Auxin alone stimulates the cell elongation, but combined with cytokinin, it make it more faster. So only auxin, it may let 10 cells to be produced.
So
Yes.
it's gonna do the opposite of elongation.
No.
Because Mr. you said when it's high, it's gonna.
Then
Reverse.
it's the same as the meristematic, Mister.
Yes, yes, it will be like as meristematic. Yes, true. Again, let me repeat it. If auxin is alone, it will cause the cell elongation. It will make the cells to divide and produce new cells, but maybe at a slower rate. If you take the same auxin and mix it with cytokinin, now the process will speed up like enzyme. Okay, so now you will have a rapid cell division and rapid growth. Okay, now the next thing which we need to see is the plant responses. We discussed that there is a stimulus, and this stimulus always causes the response in the plant. We have 3 types of the stimulus: light, we have gravity, and we have some other mechanical stimulus like touch and the contact. So If it is a light stimulus, so the response will be
Attracted
called
to
as
light. Photo.
phototropism.
Yes.
The stimulus is light, the response is phototropism.
Phototropism.
The stimulus is gravity, the response is gravitropism. What is the other name for gravitropism? Anyone remember gravitropism? What is the
Yes,
other
Mr.
name?
Downward growth.
Yes, Mr.
Geotropism. Geo mean earth. Okay, earth, that it goes towards the earth. Mechanical thigmotropism.
Thigmotropism.
Now, the most important thing, boys, over here, you have to understand that all type of the tropism can be either positive or
negative.
Negative. Like maybe one part of the plant shows a positive and the other part shows a negative. Over here in the first picture, the plant stem is showing what?
Positive, Mr.
Yes, positive phototropism, because you can only see. You don't need basically to write the positive, by the way. Okay, whenever it is positive, positive always comes alone, like in the math. So if it is a positive digit, we never write positive, but if it is a negative, we must write negative. Anyhow, geotropism, the shoots, what the shoots are showing?
Positive.
Shoots are showing
Mr. Negative.
Negative, yes.
Negative. And the roots are showing positive. Because the gravitational force pull downwards.
Yes, very good. The roots are showing positive while the shoots are showing negative. Okay. Next one, boys, here. It is what this picture is thigmotropism, because it is both towards the contact. This graph is showing the 5 plant hormones. Yeah, and it shows some of the functions over here. So you have to identify like which hormone is doing what. So it can be easily identified from this picture. Okay. So you have to make sure that you understand the hormone and you understand its function. From the table above, which hormone is not active? This is very important, boys, to focus.
Abscisic acid.
Okay, and
abscisic.
Yes, very good. If I say that from the table above, which hormone is most active? How you will get?
Mr. The most one that has correct.
Yes, very good. Okay, from the table above, which hormone limits the plant growth development the most?
Abscisic.
Abscisic acid.
How do you get abscisic acid? How you identify it?
More X's.
Yes, more X. Very good. You bought bananas and all this. So who help in fruit ripening?
Ethylene.
Ethylene.
Ethylene. Very good.
Yes.
Very good. Identify the type of tropism.
Thigmotropism.
Thigmotropism.
Very
Mr.
good.
What is A? Chemotropism.
Chemo. Okay, chemo is for chemicals. Sometime the plants, they attract towards the chemicals. Gravitropism is for the gravity. Okay. Yes. And phototropism
for
attract to light.
Light, and already the thigmo is for the touch
contact.
Or
touch, Mr. Contact.
Or
Mr. Sweet need help.
Yes, contact. Very good. Which of the below is not true about nestic?
Does not
cause movement.
Okay, so it means that nestic are reversible, nestic can be repeated, and nestic.
Mr. What is nestic?
Nestic is another type of the. You know that when we started the tropism, or when we started the plant responses, basically we divided it into 2 types: nestic and the tropism. Tropism is basically a growth response. Like you show them light, so they will show that they grow toward light or grow away from light. But in nestic, no. Nestic is another type of the movement which is not a growth response movement. You can search for this as touch me not. there is a plant that is called as touch me not. Okay, there is another plant which is
when
called
you touch it,
as,
it's close. When you touch it, it's
yes,
close.
yes. There is another plant which is called as pitcher. P-I-T, 1 minute, P-I-T-C-H-E-R maybe, if I'm not wrong. Pitcher plant. There is another plant, we call it as specifically pitcher plant. You can search these to see what is nestic movement. So when you touch the plant, what happens?
They close.
Yes.
It will
And
close, Mr. Filmov.
after some time, they will
It will
open
After
again.
some time,
Yes.
it will open again, Mr.
To collect
Yes.
the most sunlight, Mr.
Yes, so that is like a nastic response because they doesn't cause any growth response, but they are reversible. Yani you touch, they
And
close,
they are
but
can be
you
repeated.
Then they open again.
yes, and they can also be repeated. Pitcher plant is also another type of the nastic responses. You can see them. In labs, dash is added to the plant to speed up the growth faster.
Cytokinin.
Cytokinin.
Mr. Saito.
Yes. And this one is where we studied this box, this one. The question was from here. Okay?
Yeah.
When you add cytokinin, it speed up the response.
Yes,
Okay, good.
rapid cell division.
Yes. And finally, the last one. Okay. The last
Sir,
one
we have
is
so many information in our heads. We need to review. When there are so many, Mr. So many.
after this, take a break, enjoy, eat, pray, and then in the afternoon you can open this file again and quickly review. Inshallah, you will find it very easy now, trust me. Because if
Mr.
Yes, Mr. 3 hours we study only biology. Now
Yeah.
all of think it's very easy.
skeletal system, the last one, we have to just quickly see over here this KPI, which says that sequence the steps that occur during bone repair. It means that first of all, we need to know skeletal system is made up of the bones, and it helps us in support, movement, and shape. And then if by mistake or by chance or by natural thing there is a bone breakage, how the bone will repair means that there are 3 steps that happen in the bone repair. First of all, remember that the fracture will be either simple fracture, like when the bone breaks but it does not come out of the skin, it doesn't come through the skin. Then there is a compound fracture when the bone comes out through the skin, protrudes mean it comes out. And then there is a stress fracture, which means that it's like a thin crack in the bone. Okay, it's a thin crack in the bone, but it's not broken. It doesn't come out of the skin. Whenever there is something like this, we need to know the bone repair process will start very immediately. You go to the doctor put you either the cast, like the gyps, or the splint. The first step in the bone repair process is called as a fracture. No, sorry. The first step is basically when the fracture happens and you see a blood clot. A blood clot is
Sick.
okay. A blood clot is formed in the place of the fracture. So if this is the fracture area, so what will happen in the first step? The blood clot will be formed, the injured area will be swollen and inflamed, and the blood clot is formed. So this is how the first step happens. Second step is called as the connective tissue deposition or the callus formation. Okay, so we need to see over here. Sorry, 1 minute. So first step, what happens when there is a fracture? Second step,
The blood, Mr. will
cover
when the broken area.
Yes, very good. Second step, connective tissue will cover now the most of the broken area, and this is called as the callus formation step. So it takes about 3 weeks to start this process. When the osteoblast, remember there is a B, osteoblast, they form a callus, made up of the spongy bone, and later on, this spongy bone is replaced by the osteoclast. It is C now, and it is called as
It's
Muhammad.
the stronger type of.
Please, can you mute anyone's? Okay, thank you. I will repeat this step again. Second step is the callus formation. When the connective tissue covers mostly the broken area, it forms a callus, and this callus is mainly formed of the osteoblast. And then osteoblast, they make a soft bone or the spongy bone. But later on, this spongy bone is replaced by the osteoclast, and they make a compact bone. Now, the last one is called as remodeling. Remodeling is giving it a shape back. You have a healed fracture, and remodeling basically there is not a specific time. It depends on nutrition, age. It depends on age.
Lack of calcium.
Yes, it depends on many other factors like how strong the bone was broken, where the bone was broken, in the hand, in the
Your
backbone.
age.
Yes, age as well. Yes. So the person have to eat the things which are good for the bone repair.
A lot of calcium, sir. It is a lot
Yes.
of calcium.
Yes. So they need to eat more calcium. Young people will heal the bone faster as compared to the old people, and it may take from like quick like 1 month to like 1.5 month, even 2 months, 3 months, sometimes 6 months. Okay, so it depends. Is it clear, boys? So the 3 steps.
Yes.
Yes, sir.
The bone is broken, then covered by the blood clot, then connective tissue replace the blood clot, and then the remodeling happens. Okay. This question: red and white blood cells are produced in where?
Red bones mirror.
Red
Red
bone.
bone marrow.
Very
Bones
good.
marrow.
New bone growth and the bone repair. You go up and see this is the bone repair process. This one, the bone repair process. And who is responsible for the bone repair? Osteoblast. Very good. Yes, thank you. Osteoblast. Explain the steps in the bone repair. So what are the steps in the bone repair?
Collect
Red
blood
first.
to form in the space between the broken bones.
Yeah,
The
so
first
number
step.
1, clotted blood forms in the space, right?
In the space between the broken bones.
Yes, space between the broken bone. Okay. What is step number 2?
Connective tissues
number
then
2,
fills the space between the broken bones.
Yes, connective
Mister,
tissue.
it will not connect it all, Mister. Most of it. It will connect most of it.
Yes, yes,
Not
very
all.
good. Yes, very good. It fills the space between the broken bones. Okay, between the broken bone. Okay, and number 3,
Remodeling.
Osteoblast produce
Yes,
new
sir.
bone tissues.
yeah, where we say that the osteoblast, the question number 2, basically, osteoblasts produce
Will
a new
repair.
bone tissue.
A new growth.
bone tissue and do the bone repair. Okay, good. So this is how it works, boys, and that's it. You deserve from me a big thank you that you stayed here for so long. So thank you so much.
Mister,
And
we should say
Yes, Mister. We should to thank you.
Yes, Mister. We're the
Thank
one
you,
that
Mister, for
thank
your
you,
time.
Mister.
3 hours.
wish you all the best for the
Thank you,
exam.
Mr. Muhammad.